Good morning, dear students. We have today a topic called as metabolism of purine nucleotides. Previous lectures we were dealing with metabolic products such as citric acid. We went along with a further discussion regarding various different pathways like glycolysis, the ED pathway, pentose-phosphate pathway, and now we are at the end of the syllabus part that is called as metabolism of purine nucleotides. So the process can be divided into two phases: synthesis of amino imidazole ribosyl 5-phosphate from ribose 5-phosphate through 5-phosphoribosyl 1-pyrophosphates (PRPP) it is called as, and second will be synthesis of inosine monophosphate from amino imidazole ribosyl 5-phosphate. So these are 2 points wherein we can directly work out in this particular fashion. Now, biosynthetic pathways of purine nucleotides. So if you see, right from D-ribose 5-phosphates, the synthesis process takes place with various different metabolisms or different intermediates of metabolism, and ultimately the synthesis of structure of that particular purine molecule takes place. So the pathway of de novo purine biosynthesis from ribose 5-phosphates and ATP starts with transfer of pyrophosphates from ATP to C1. See here, now this is alpha D-ribose 5-phosphate with the help of enzyme PRPP synthetase Mg2 plus ions ATP to AMP, and ultimately PRPP remains as it is and additional phosphate group will be added. In the second reaction, if you see, from the additional phosphate group with the help of an enzyme PRPP glutamyl aminotransferase, glutamine is converted to glutamate and the 5-phospho beta D ribosyl amine is being generated. So if you just consider the reaction, the reaction involves inversion of configuration of C1 and forms what will become beta N glycosidic bond. So formation of beta N glycosidic bond takes place, and ultimately this product, that is 5-phospho beta D ribosyl amine, is being generated. Condensation of glycine to glycinamide ribosyl 5-phosphate. So this addition would take place and generation of glycinamide ribosyl 5-phosphate would occur. Then from there, glycinamide ribosyl 5-phosphate, there is formal transferase kind of enzyme by which there will be transfer of formyl glycinamide ribosyl 5-phosphate. See here, glycinamide, the formation, the transfer of formyl group takes place at this particular position and the product is being generated. Now, formyl glycinamide glycosyl 5-phosphate gets converted to formyl glycinamide ribosyl 5-phosphate. Elimination of water accompanied by ring closure from aminoimidazole ribosyl 5-phosphate. The initial event is phosphoribosyl group transfer from ATP to oxo functions, nucleophilic attack. Then amino imidazole ribosyl 5-phosphate gets now converted into amino imidazole carboxylate ribosyl 5-phosphate. So this is another intermediate compound that is being generated because of the reaction which is catalyzed. Amino imidazole carboxyl 5-phosphate formation would take place in this reaction. Furthermore, amino imidazole carboxylate ribosyl 5-phosphate now gets converted into amino imidazole succinyl carboxamide ribosyl 5-phosphate. The group of succinyl group is being attached over here. Ornithine and arginine in the cycle, condensation of aspartate to form amino imidazole succinyl carboxyl ribosyl 5-phosphate would take place. Correct. So this is how the actual reaction works out. Now, in this reaction, if you see in the early stages, right from here, if you see, there are various different enzymes which are interacting in this particular fashion, and these reactions or these enzymes are said to be enzymes which are helping in the metabolism of purine nucleotides. Correct. For example, PRPP synthetase is there, which is the very initial or the first enzyme of the particular pathway. Second value is PRPP glutamyl aminotransferase. Third one, the formation of ATP molecule will be generated or it will be utilized here in the presence of glycine. Formyl transferase the enzyme it is. Again, continuing with the thought, and ultimately adenosine succinate. So actually today we have seen only up to this particular part. In the next lecture we will try to commence with these reaction as well, and the closed ring structure of the purine molecules would take place. Correct. So based on these reactions, the purine molecules will be there, and the configuration or the structure of the purine molecules will be worked out very correctly. Correct. So we will be stopping here only, because the reactions that we have conducted right from this particular reaction of PRBP synthetase, addition of the glutamine and conversion of glutamine to glutamate with the help of H2O molecules, utilization with the help of enzyme PRBP glutamyl aminotransferase. Then there will be the 5-phosphobeta-D ribosamine gets converted to glycinamide. So glycine molecules get attached to it in order to make a proper configuration. Formyl group is being generated with the help of enzyme formyl transferase. See this. Then the glutamine again converted to glutamate, addition would take place. Amino imidazole ribosyl 5-phosphate, and this gets converted to amino imidazole carboxylate ribosyl 5-phosphate. Then it is converted with the help of an aspartate which comes in play. It gets converted to amino imidazole succinyl carboxyl ribosyl 5-phosphate. So these are all catalysis reactions that takes place with the help of different enzymes. So, thank you.